Okay, in this lesson, I just want to reiterate basically what I said in the previous lesson and hopefully just to make things a little bit more obvious for you guys. So in this case, we have another crossing between a male and a female, but in this case, it's not plants. We can look at what happens in rats, just as an example. And here we're also using the Punnett square. So basically, when we get the results for the offspring, and again, your possibilities of what type of offspring you're going to have, they can ask you to do it one of two ways. Either give your results in a Punnett square, where you literally use like actually a square to figure this out. It's quite easy if you know what you're doing. And there's a second method as well. So after this slide, we're going to talk about the second method. That's just called your genetic diagram. So they can either ask you to give the answer in a Punnett square like this. Again, you don't have to draw pictures or anything. It's just a quicker way of doing it, like in the previous lesson. In this case, they just show you the pictures for reference sake. You don't have to draw anything. It's just a Punnett, and you just write your letters in. So either the Punnett square, or you can do it in the genetic diagram method. Usually the genetic diagram method gives you the most marks, and it's actually the easiest, again, if you understand what to do. Okay, so let's just quickly go through this guy. In this case, you have rats. Okay, so you've got a male in this case, and you've got a female. In this case, they show you again, each of these two have 2 chromosomes. Okay, for this female, she got one of her chromosomes from dad, and she got another chromosome from mum. Okay, so this is a homologous pair. And on these 2 chromosomes, she has a specific gene that encodes for, in this case, color. So in this case, the trait, the characteristic we're going to look at is color. Okay, so both these rats happen to be gray in color. And also, it happens that both these male and female, the rats, they've got the same alleles. So both of these are heterozygous, capital B, small b. So in this case, the B represents the color. In this case, it's going to be gray. Okay, so because they're heterozygous, they are gray in color, capital B, small b. In this case, it's another individual, capital B, small b. So both of these rats are physically, phenotypically gray, and genotypically, they are heterozygous because I've got a capital B and a small b. Okay, so I hope you guys are with me so far. You have to remember these terms because you have to use these terms when you do this type of crossing. Okay, so in hierdie geval, het is 2 individue, manlik en vroulik 2 muise. En in hierdie geval kyk ons na 'n ander karaktertrek, ons kyk na die kleur. Hierdie geval toevallig is albei muise grys van kleur, fenotypies. So hulle vra vir die fenotype van die ouers, dan weet ons, en hulle vra vir kleur, dan sê jy net hulle is grys. So fenotypies is hulle grys. Genotypies, as ons kyk na die gene, is hulle heterozygoties. Ons sê hoofletter B en 'n klein b, hoofletter B en 'n klein b. So albei is heterozygoties toevallig. Okay, en weer eens wat hierdie kromosome beteken. Elke individu het 1 kromosoom van kom sê maar pa, die eerste een, en 1 van hulle eie ma, en dit maak dat hulle heterozygoties is. Toevallig, daar is nie dieselfde alleel op albei die kromosome nie. Elke alleel in die geval het 'n unieke alleel, so dit maak hulle heterozygoties in hierdie geval. Hoofletter B, klein b. Weereens, hoofletter beteken is dominant, kleinletter beteken hierdie enetjie is resessief. So in hierdie geval kan ons eintlik sien dat grys die dominante kleur is. Hoekom? Want dis een alleel wat die effek van die ander een gaan verdoesel, en ons sien dat hierdie rot grys is. So ons kan aanneem dat die kleur grys is dominant oor wat ook al ander kleure dit is. Ons gaan sien dat dit bruin is, soos in hierdie geval. So grys is dominant oor bruin. Okay, just quickly for the English. In this case, we can see both these individuals are gray in color. But we can also see that since they're heterozygous, there's a capital B and a small b. And remember, we said usually your capital letters are dominant over your small letters. And the fact that this individual is gray in color, and it has a dominant and a recessive allele, this tells us that the dominant allele must be there for gray. So the fact that you have, or this individual has a dominant allele, the dominant color then must be gray. Because if this one then was for brown, the individual would only be brown if it was homozygous recessive. Like in this case, here we have a possibility of an offspring that is brown, but it must have both recessive alleles for you to see phenotypically the brown color. Okay, so go on to the crossing. So then, with after meiosis for your female, 2 egg cells. When these 2 chromosomes, this just shows you again physically what happens. When these 2 chromosomes split from one another after meiosis, after metaphase, one chromosome goes into one egg cell, the other chromosome goes into the other egg cell. Okay, so here are the 2 possibilities of eggs. One will have a capital B, one will have a small b. They split up from one another. For the male, after meiosis, these 2 chromosomes does the same thing. They split from one another. One goes into one sperm cell. The other one goes into another sperm cell. So here we have a capital B along with all the other chromosomes in this sperm cell. And here we have a small b in this one. Okay, so far so good. So wanneer hierdie gamete van mekaar of die kromosome van mekaar onderskei, van mekaar geskei word gedurende meiose, hier is een eierselletjie met 'n hoofletter B in die kromosoom, daai alleel, en dan hierdie eiersel het ons die klein b op hierdie alleel van hierdie kromosoom. So hulle word fisies van mekaar geskei gedurende meiose. Dieselfde gebeur met die manlike individu. Gedurende meiose vorm hy spermgamete en een van die gamete gaan 'n hoofletter B wees en die ander enetjie gaan na 'n klein letter B wees met die kromosome skei van mekaar. So then, if you have your Punnett, you have your capital B, small b over there. And now you just fuse them together because what's happening inside the Punnett is fertilization. So this entire Punnett, or this square, represents fertilization. So when this sperm cell meets this egg cell, you have an individual that has a capital B and a capital B chromosomes, alleles. And this guy then is homozygous dominant. And phenotypically, it's a grey individual because it can only be grey because it's got 2 capital B's, and like we said, the grey color then is dominant over brown. So genotypically, it's capital B, homozygous dominant, but phenotypically, it's grey. Okay, so then again, if we have this sperm that fertilizes this sperm, we have a heterozygous individual. So you just bring this B down and this B to the side. So these 2 chromosomes then meet. So you have a capital B and a small b. So this individual genotypically is heterozygous, but phenotypically, physically, is also grey. Why? Because we have one capital B and this guy suppresses the effect of this one. So it's kind of like this little B is not there for us to see on the outside. So just by looking at this rat, it looks the same as this one. We will never know whether it's homozygous or heterozygous without a DNA test. Only with testing the actual genes will we be able to see, okay, its genotype is heterozygous in this case. But just by looking at the color, you won't be able to know it. It's going to look exactly the same on the outside as this one over here. Okay, so then again, if you have your capital B, the sperm, meeting this egg cell, you also get a heterozygous individual, also possible. But if this sperm fuses with this egg cell, you're going to get phenotypically a brown rat and genotypically a homozygous recessive individual, 2 small b's, when these chromosomes meet. So now we can see, okay, it is a brown rat, so genetically there must be something different inside this rat. So it's homozygous recessive. Okay, so vandag wanneer hierdie spermsel weer met die eiersel ontmoet, kry ons dan 'n homozygotiese dominante individu, genotypes, maar fenotypies gaan dit grys wees, want dit is 'n dominante alleel. Hy verdoesel daarom niks nie, so daar's 2 van dieselfde allele, dit kan net grys wees. Die spermselletjie en hierdie eierselletjie gee dan vir jou 'n heterozygotiese individu reg hier. So genotypies is dit heterozygoties, hoofletter B, klein b. En fenotypies gaan hulle grys wees. Hoekom? Want hierdie hoofletter B verdoesel die effek van hierdie klein b. So dit is asof die klein b nie daar is nie. So omdat hy daai dominante een het, gaan dit dan grys wees. So dit is nou volledige dominansie. So dit wys dan net vir ons grys. Die dominante een is die een wat ons fisies gaan sien aan die buitekant. So ons gaan eintlik nooit weet wat die verskil is tussen hierdie 2 rotte nie, want dit is albei grys. Ons gaan nie weet wat hulle genotipe is tensy ons 'n genetiese toets doen nie. Dan gaan ons sien, okay, die allele wat hy het, is 2 verskillende allele op die DNA van hierdie 2 allele of hierdie 2 kromosome, gaan dit dalk 'n mutasie wees wat hom hierdie ander alleel gee. Maar fenotypies aan die buitekant is hy presies dieselfde, hy's grys. Net hierdie enetjie in die hoek, wanneer hierdie spermselletjie en hierdie eierselletjie mekaar bevrug, kry ons die moontlikheid van 'n breinrot fenotipies. Genotipies sien ons dat hy homozigoties resessief is, want weer eens is 2 van die rare allele wat bymekaar kom en hierdie 2 bymekaar gee dan vir jou 'n breinrot. Okay, and what these quarters mean is that you've got a 1 out of 4 chance of getting a homozygous dominant individual. You've got, technically, when you plus these two together, so it's 2 out of 4, you're going to get a 2 out of 4 possibility of a heterozygous one, because literally there's 2 heterozygous individuals here, but it's a chance. So 2 out of 4 chance of getting a heterozygous individual as a child. And there is a 1 out of 4 possibility for these 2 parents to have a brown offspring. So again, the Punnett square is all about possibility, not about the fact that they're going to have 4 children. They could just have 1 child and then never have any more. But there's if they get the 1 child, there's a 1 in 4 chance for it to look like this, 1 in 4 chance for it to look like this, or a 2 out of 4 chance of looking like this. Okay. So hierdie Punnett vierkant gaan dit alles oor die moontlikheid, die moontlikheid vir een van as hulle een kind het, wanneer hierdie 2 rotte een kind het, is daar 'n 1 uit 4 kans dat die kind so gaan lyk, 1 uit 4 kans dat hy so gaan lyk en 2 uit 4 kanse dat hy heterozigoties gaan wees. Tog grys, maar vir heterozigoties. So fenotypies 3 uit 4 kans dat dit grys gaan wees. Phenotypies, 1 uit 4 kans dat 'n bruin gaan wees. So guys, and the chances of it for the phenotype, having a grey child, for these 2 individuals having another grey baby, is 3 out of 4, because look, there's 3 possibilities for it to be grey, but there's only a 1 out of 4 possibility that the offspring will be brown. Okay, so it's all about possibilities, genotype-wise and phenotype-wise. Okay, then just quickly, the genetic diagrams. I think this will go more quicker, because it's basically what we just did in the previous one, but we're not using a square. Okay, so in this case, they tell you in words. So this you'll have to literally write out. So in this example, they tell you that the P1 generation, or your parents, you've got a heterozygous grey male. So we're just doing the same one as the previous slide. So we have a grey male mouse, but it's heterozygous. And it is crossed with a heterozygous grey female. Okay, so basically what we saw in the previous one. So how are we going to show this? We always have to write phenotype and genotype. Okay, so we just had the phenotype. This one's grey, other one's also grey. Brilliant. For the genotype, they said it's heterozygous. So they will tell you what to use, what letter. But let's use the letter B again, like in the previous activity. This is heterozygous, capital, small, and it mates another heterozygous female, capital, small. Okay, hope you're all with me. Then what happens is Moses. You have to write out Moses. So meaning this entire slide is what you guys will have to write out yourself. This question can be easily 7 marks, 8 marks, tops. So you have to write out all these steps. The fact that you write that meiosis happens will also give you a mark. So don't forget this step. Meiosis happens. So meaning this allele will split into its own gamete, and this allele will split into its own gamete. Same thing for this individual. So here these circles represent the gametes, but this is still from your male. This one is from your female. So you have to show, you can still cross. So you have to show the X. Then fertilization can happen. So again, for your F1 generation, your first offspring, there's going to be some genotypes and there's going to be some phenotypes. So genotypically what can happen? This capital B, this sperm cell, can come down and be here. Then it can fertilize with the first egg cell of the female, possibly. So we write it here. So you can have a grey individual that is homozygous dominant. Okay, so again, same answer as in the previous slide. We're just not using any pictures in this case or any punnets. Okay, but what can else happen? This gamete can also fuse with the second egg cell of this individual. So then we have a capital B that will be together with a small b. And yes, you guys will have to write these lines out. This is actually for you to remember what have I paired with what. What do I still need to pair with what? And for the examiner to see, okay, you know how the fertilization possibilities can happen. Okay, so you can get this type of individual, you can get a heterozygous individual, then what if this small b gamete fuses with the first one from this, capital B, small b, and we usually write the capital before the small. If you turn it around, no worries, you'll still get your mark, but I like to put the capital in front of the small letter. So this one you can see is also heterozygous. But this small b can also fertilize with the last one over here, giving you a homozygous recessive individual, also possibility. So what are the phenotypes? You can get a gray, a gray, a gray, and also a brown individual based on this crossing. And again, it's not for children. This shows you that there is a 1 out of 4 chance of getting this individual, a 2 out of 4 chance of getting this individual, and a 1 out of 4 chance of getting this individual for the genotypes, okay? Genotypes, what the alleles are like. They can also ask you what are the chances for the phenotype. There's a 1, 2, 3 out of 4 possibility of getting a gray individual and a 1 out of 4 possibility of getting a brown individual. So also what you have to write down here for your genetic diagram at the end is your genotype ratio and your phenotype ratio. So again, this is basically what we just said. There's one individual that's capital B, so the ratio is 1 to 2 individuals that is heterozygous to 1 individual that is homozygous recessive. So the ratio technically is 1 to 2 to 1. But always write down your genotype so that the examiner knows that you know what the 1 represents and the 2 represents and this 1 represents. So phenotypically, what again here is the ratio? 3 to 1. 3 could be gray and only 1 possibility of a brown individual. Okay, so when you guys study again, please study this slide. I just quickly want to show you guys in the last slide what can happen if we go to the second generation. Okay, so let's kies dit gewoon Afrikaans. So wat gaan gebeur as hulle vir jou 'n heterozygotiese grys mannetjie gee en ons gebruik nou weer die voorbeeld van die rot en hy paar met 'n heterozygotiese grys wyfie. Al hierdie synotaatjies byskryf. Jy kry punte as jy noem wat gebeur en dan moet jy hier fisies wys wat gebeur met die gamete wanneer hulle paar mekaar. Okay, so fenotypies het ons 2 grys individue wat gaan paar. Genotypies is hulle al 2 heterozygoties. So jy moet dit wys plus jou X wat wys hulle gaan mekaar paar. Dan gebeur die proses van meiose en dan kry jy jou gamete. So hier weer eens, hierdie hoofletter B skei van die klein letter B. So 2 moontlikhede van gamete vir jou mannetjie. Dan vir jou wyfie het jy die moontlikheid van 'n hoofletter B gameet en 'n klein letter B gameet, want hierdie 2 skei van mekaar. Okay, na meiose. Dan vind bevrugting plaas. So vir jou F1 generasie, jou volgende kindergenerasie, gaan jy genotipes kry, moontlike genotipes en moontlike fenotipes. So die moontlike genotipes hier gaan wees van hierdie enetjie verbind met die eerste een van die volgende individu. So jy kan 'n homozygotiese dominante individu hier kry, of hierdie selfe gameet kan ook bind met die laaste een van die volgende individu van die vrou. Dan kry jy heterozygotiese een. So weer eens, jy moet die lyne wys, en die lyne wys vir die eksaminator dat jy weet wie het met wie gepaar. So daar gaan elke keer 4 moontlikhede wees van kinders wat uitkom, van jou monohybride kruising. So guys, just again, this is a monohybrid cross. We're still just looking at one characteristic. I stress this because we are going to look at a dihybrid cross later on, when we look at more than one characteristic. So for monohybrid, we always will have 4 possibilities. Vir jou monohybride kruis gaan ons altyd 4 moontlikhede hê. Okay, so weer eens, as hierdie gameet sê nou maar met die eerste een van hierdie paar, dan kry ons ook 'n heterozygotiese individu, maar hierdie ouetjie kan ook paar met die laaste een van die ouetjie. En dan kry ons 'n homozygotiese resessiewe een. Okay, so weer eens, wat is jou verhoudinge? Aan die einde moet jy hier skryf as jou verhoudinge. Kom ons begin met die genotipe. Daar is 'n 1 kans, 1 uit 4 vir 'n homozygotiese dominantskies en hy is 1 BB. Jy kry 1 moontlikheid van hom. Maar daar's 2 moontlikhede, 2 van hierdie heterozygotiese een en dan 'n kans uit 4 om so enetjie te kry. So jou verhouding basies is 1 tot 2 tot 1, maar skryf asseblief jou gamete neer sodat jy kan onthou waarna verwys die een? Hierdie verwys na die eerste ouetjie. Daar's 2 wat so lyk en daar's 1 wat so lyk in hierdie kruising. Fenotipies gaan daar 3 grys wees en 1 bruin. 1, 2, 3 grys tot 1 bruin. Dis jou fenotype. Dit is fisies in die buitekant sien. Okay, then just lastly, this is the second slide. So what if, for P2 generation, we were to take, okay, let's just go to the previous slide, sorry. Remember, we had a heterozygous grey male, heterozygous grey female, and these were the possibilities of the children. But in the next slide, I want to show what happens if we were to say cross, let me just quickly see, oh yeah, a heterozygous offspring, and we were to mate this male with its sister that was brown. We saw on the previous slide there's a possibility for a brown one. Okay, so grey and a brown individual. So here we have a heterozygous grey male that mates with a homozygous brown female. What possibly can happen? I'm not going to go through everything in detail again. Again, you have to write down all the possibilities here to the side. But I want to show you what happens, what's going to be different. In this case, everything's going to be the same. Same gametes will be formed because you have a capital B and a small b. But for the brown, we're going to have 2 small b's in the gametes. Because remember, to have a brown individual, it must be recessive, homozygous recessive. Meaning homozygous, 2 of the same. But it's recessive to give you brown. It's the rare type. Okay. So in this case, what's not going to differ? Your ratio is going to differ. So the first possibility, we get a heterozygous 1. For the second possibility, we also get a heterozygous 1. Here we have a homozygous 1, small b, and also this small b can pair with that small b. So what are the ratios now? It has differed. 2 of the individuals, 2 possibilities can be heterozygous. 2 possibilities can be brown. So there's a 2 out of 4 chance of the individual being heterozygous. And there's a 2 out of 4 chance of the individual being homozygous recessive. There's also a 2 out of 4 chance for the individual to be grey. And there's also a 2 out of 4 chance for the individual to be brown. So from this crossing, these 2 parents, it's possible to get a grey individual, and it's possible to get a brown individual. Okay, so the only possibilities for this crossing is that these parents can have either a brown child or a grey child. Okay, 2 out of 4, so it's kind of 50-50. There's a 50% chance for the individual to be grey, 50% chance for it to be brown. Same thing with the genotype. 50% chance for grey, 50% chance for heterozygous, and 50% chance for homozygous recessive. Okay, so nou vinnig wat die verskil is, as ons nou vat een van die heterozygotiese grys mannetjies in die vorige kruising, het ons gesien as 'n moontlikheid van so 'n individu, 'n kind. As hierdie kind nou met sy suster gepaar word, wat homozygoties bruin is, wat sou gebeur? So nou het ons 'n grys individu en 'n bruin individu wat mekaar paar. Die gryse gaan weer heterozygoties wees, hoekom hulle sê vir jou hy gaan wees. En die bruin een is homozygoties, want ons het beide van hierdie resessiewe allele nodig om vir jou 'n bruin individu te gee. Die gamete gaan 'n bietjie verskil. Hier is 'n hoofletter B, klein b, want hierdie word geskei tydens meiose. Maar hier het ons 2 van dieselfde gamete. Jy kan net 'n klein b gameet kry, want dit is al tipe alleel wat die ouetjie het. So dan, hoe gaan dit nou verskil? Hier het ons 2 heterozygotiese individue, as jy die lyne volg, en dan die 2 homozygoties resessiewe individue, moontlikhede, as jy weer die lyne volg van hoe die gamete met mekaar kan bevrug. So, ek wil wys dat die verhouding hier gaan verskil. So in hierdie kruising sien ons dat daar is 'n kans vir 2 heterozygotiese individue en 2 homozygoties resessiewe individue. So die kanse om 'n bruin individu te kry uit hierdie kruising uit, is 'n kans van 2 uit 4 uit. So jy tel net hierdie met mekaar. 1, 2, 3, 4. 2 van die 4 is bruin. So daar's 'n 50% kans vir hulle om 'n bruin kind te hê. Dan is daar 'n 2 uit 4 kans vir die individu om ook grys te wees. So 'n 50% kans dat die kind gaan bruin wees. Grys wees. Bruin en grys gaan wees. En dan is daar 'n 50% kans dat hy heterozygoties gaan wees. En daar is 'n 50% kans dat hy homozygoties recessief gaan wees. Okay. So I'm going to stop with you guys right here. A lot of homework is definitely necessary for practicing these types of crosses.